Everybody on Zoom hear us okay? Give us a thumbs up. Okay, great. Well, thank you all so much for being here, either in person or on Zoom. I just want to welcome you to Chris's thesis defense. I've been so excited about this all week. I know Chris has been super excited. For those of you who don't know me, my name is Lori Svoboda. I'm Chris's thesis mentor. I've had the pleasure of mentoring him over the last 2 years, alongside some outstanding colleagues, Andre Monteiro da Rocha, Todd Heron, who are both at the Cardiovascular Regeneration Corps, and we've been working very closely with them on this work, and that's where we've been conducting the research at this point. And also Dr. Justin Colacino here in EHS. And just to give you a little background, I met Chris three years ago, actually, when he was still trying to decide where to go for graduate school. And I was just blown away by how smart, how well-spoken he was, and how passionate he was. I mean, he clearly had so much passion for environmental health, and in PFAS in particular, growing up in a community that was highly exposed to PFAS. And I gave him an excellent evaluation, and I said, we have to get this student to come to Michigan because he's amazing. And then I was also a little bit bummed because I didn't think he was terribly interested in basic science research at the time. I was like, wow, it'd be amazing to have him work with me, but I know he's not going to work with me, but oh well. And so I was absolutely delighted when he came to me just a year ago. Was it just a year ago? I can't believe it's just been a year, but he wanted to do some work just as a paid work study student on some of the research that he'll be talking about today. And that turned into his interest in doing a master's thesis with me, which again, I was so delighted. And he's just blown me away with his, how quickly he has caught on to things, very difficult techniques, including culturing stem cells, differentiating them into cardiomyocytes, taking those cardiomyocytes and culturing them, mapping calcium and sodium currents in those cardiomyocytes, analyzing complex data sets. So you'll get to see a lot of that work presented today. So just know that all of this has happened in less than a year, and we have a manuscript that we're going to be putting together in short order. And he has also been, he's been quite a leader in the laboratory there with a lot of the undergraduate students have started to look up to him, and he's mentored a lot of them and taught them some of these techniques. He's been a wonderful friend and colleague to all of the students in the Do Goods lab and in the EHS department. And I know that he's going to go on and do amazing things. I'm very happy that he's not leaving just yet. He's going to stay on with us in the lab and do some more of this work and keep moving these projects forward while he figures out the next phase of his life. And yeah, I'm just so excited to see where you go next and where your master's takes you. And so with that, I'll turn it over to you. Thank you so much.
Pretty surreal that we've already reached this point in the program. I've reached this point in the program, this point in the year, and I'm super grateful to see so many friends and family here on Zoom and in person. And so without any further ado, I'd like to start with my thesis presentation, which is the effect of perfluorooctanoic acid and hexafluoropropylene oxide dimer acid on the function of human-induced pluripotent stem cell-derived cardiomyocytes. Try to say that 3 times fast. So with a little bit of an introduction, I'll start with polyfluoroalkyl substances. And what is the origin story? Why is this kid so obsessed with PFAS? So this is my hometown, Westfield, Massachusetts, located in Western Massachusetts. And where that star is located is an Air Force base. And at that Air Force base, they utilize something called aqueous film forming foam, which is a firefighting foam used by, oh, you know what? I realize I'm not sharing my screen. So I'm glad I caught that. I'm sure people on Zoom were like, can you share your slides? Yep. Okie dokie. There we go. I'm glad I caught that. Yay, for people on Zoom. see that? I think they can. Can I get a visual confirmation from somebody? Awesome. All right, we're good. Great. Sorry about that. Glad I caught that. So I'm from Westfield. This is the Air Force Base. They use aqueous film forming foam, also known as AFFF, which contains a complex mixture of different PFAS compounds. And that Air Force Base is located on top of our city's aquifer, where we drank that contaminated water for almost 40 years. And you can imagine, I'm pretty pissed. So the Air Force originally developed this chemical in the 1950s and utilized it up and starting in the 1960s. And in Westfield, they were using it at the Air Force Base in the 70s. So in 1974, the Air Force started recognizing the toxic effects on AFFF of fish. In 87, in Massachusetts, the Mass Department of Environmental Protection ordered a cease and desist order. The DoD did not stop using AFFF foam, AFFF. In 2000, they realized that certain PFASs are toxic and carcinogenic. And then in 2013, so between 1987 and 2013, we were getting that contaminated drinking water. And in 2016 is when those taps were finally turned off. So almost 40 years of exposure. And what the heck am I going to do with that? So in reflecting on this, I realized that my condition of being exposed to this chemical was not unique. Let me get rid of this. Is not unique. PFAS exposure is unavoidable. You can find it in nonstick cookware, furniture, clothing, all of these this wide variety of common household use products. And just here's a short list of the industries and use designations for PFAS. And there's the rest of them. So, and those are just the ones we know of right now. And this is for the wide class of chemicals. So PFAS is a concern of is a chemical of concern of global contamination. Here in the United States, these are all the sites that we currently know where PFAS contamination exists. And Michigan actually has the most known sites of PFAS contamination. You can imagine though, where those marks are, is just where we've looked for it. And I'm sure one day, once we really characterize PFAS contamination in the United States, this entire map will be blue, purple, and orange. Not only concerning that is that this chemical can be found in the Arctic, and there is no human activity happening in the Arctic. So somehow this chemical is being transported over long distances, and it's highly mobile in the environment. It's resistant to degradation, and because of that, it has the potential to bioaccumulate. And the chemicals that I'm interested in, they have biological half-lives on the order of magnitude of years. And so you can imagine that when you have 10 to 12,000 different ones, that can be really concerning from a regulatory perspective, because you're trying to regulate and control these chemicals, but when you have to do that for 12,000 chemicals, that's no easy task. So the two chemicals of interest in this study are GenX, which is the contemporary PFAS compound that people are regularly exposed to today. However, we're still exposed to PFOA, which is the legacy contaminant that was originally generated, and GenX has been used as a replacement for PFOA. So in the United States, thankfully, there's been a decreasing trend in PFAS concentration in human blood. With the coordination of industry and the EPA, there was a voluntary phase out that led up till 2015. And so thankfully, we've seen a decline. However, these concentrations can still be detected in 99% of Americans. It may also be of note that workers that were working in facilities that were manufacturing and using these chemicals in the production of products have the highest burden of chemicals in their bodies, 3 orders of magnitude, greater concentration than the average population. And so PFAS is associated with a wide variety of adverse health effects, which is why we're so concerned about it. It's been associated with developmental effects, endocrine disruption, liver disease, testicular cancer. There's a wide variety of different effects depending on which chemical structure you studied and how long you've studied it. And now to kind of pivot a little bit from PFAS, I also want to talk about the heart's function and disease. So we have the human, and inside that is our cardiovascular system, which is comprised of the veins, arteries, our heart, And below that, within the heart, is the cardiac muscle tissue. And inside of that cardiac muscle tissue is this mix of different cell types, including cardiomyocytes, which will be the cell of interest, endothelial cells, fibroblasts, and smooth muscle cells. And there's a few other ones I didn't mention. And in conjunction with the cardiomyocyte are these myofibrils and their filaments. And once you get lower than that, you have organelles. And once you get lower than that, it's molecules, and then it's atoms, and then quarks. And we could go all up and down on the orders of levels of organization. But this is where I want you to keep your brain for the rest of this discussion as we start thinking and talking about cardiovascular disease in relation to environmental exposures. So electrophysiology of the heart, it's comprised, the heart is comprised of these excitable cells, and they promote beating through the coordinated action of action potentials. So there's my little cardiomyocyte here. This is probably a nicer picture, but this is my fun PowerPoint cartoon of one. But as you can see, they're kind of connected together by these gap junctions. And that's where the signal from one cardiomyocyte jumps to the next cardiomyocyte to say, okay, it's time to contract. And so these action potentials are generated because of the movement of ions, including calcium, potassium, and sodium. And these movements the action of these cells is coordinated through the pacemaker in the heart, and it excites cardiomyocytes. And you can see here, this is action potential structures, and the shape of that action potential is going to be different depending on what location of the heart you're in. And so for this study, we use isocell cardiomyocytes, but we also use ventricular cardiomyocytes. So we're more interested in this bottom shape, or this is the shape of a lot of our peaks. So within the cardiomyocyte, there's a large variety of potential sites of toxicity. So here's another diagram of the cardiomyocyte with different ion transports and its organelles. And so substances that cause toxic effects do so by influencing heartbeat, heart rhythm, contractile strength, so the actual kind of undulation of the heart. And the heart is a really sensitive organ for toxicity and of damage because of its limited proliferative capacity. If you were to take the mass of your heart, about 2% of the cardiomyocytes in that mass are replaced every year. So you can imagine if you were to sustain cardiac damage, it's going to take a long time for your heart to get back up to a capacity where it's actually regenerated to functional capacity. And unfortunately, it probably will never be to the same level of functional capacity prior to the cardiac injury. So there's a few different sites of action where our chemicals of interest may be at play, including these ion channels and ion pumps. You can think about the membrane and the receptors bound to the membrane, the microsomes. Mitochondria is a huge source of interest because that's where, as you all potentially know, that is the powerhouse of the cell. But did you know that it is also one of the main storage houses of calcium in the cell? I don't think they ever really tell you that one. And then there's intracellular gap junctions and the sarcoplasmic reticulum, which is the primary storage house of calcium inside of the cells. So now we connect. Now we're going to start pairing PFAS and the effects that we see in the heart together. And so cardiovascular disease overall is the leading cause of death in the United States. 1 in 4 deaths is associated with cardiovascular disease. And again, this disruption can happen because of biochemical, through biochemical damage and disruptions in the cellular pathways and function of the cell. Manifestations, there's a wide variety. Cardiovascular disease is like a huge bridge of arrhythmias, hypertrophy, cardiomyopathy, coronary disease, overt heart failure. Those are just fancy terms for different functional endpoints that can affect the heart that lead to death. And we know that environmental exposures have been associated with cardiovascular disease. If you look at the incidence of strokes and heart attacks on days where there are high amounts of air pollution, we see an increased risk of heart attack and stroke. PFOA and other PFAS exposure has been kind of somewhat associated, but because there isn't a strong enough association, the ATSCR, when they conducted their survey of toxicological evidence, stated that there isn't sufficient evidence to say that PFOA exposure increases the risk of heart disease. And when we're thinking about, now to finally bridge the gap, when we're thinking about cardiovascular disease, there's actually a lot of risk factors and comorbidities that are involved in the generation of that, or the ontogeny of cardiovascular disease, which may include low birth weight at birth, changes in cholesterol, type 2 diabetes, adiposity, and obesity. And what's interesting, sorry, wrong direction. And also disrupted electrophysiology, which can lead to like an arrhythmia, for example. What's really interesting though too is that PFAS exposure has also been associated with the comorbidities that may precipitate cardiovascular disease, as it's been associated with low birth weight, changes in cholesterol, type 2 diabetes, and adiposity. Now, one of the questions I have though, can we build this direct bridge between PFAS exposure, disrupted electrophysiology, precipitating cardiovascular disease? Because right now there's kind of this cloud of effects that are like ancillary to each other, but can we draw this causal pathway of disrupted electrophysiology? So now let's go into the project goals. So I had two aims of the study. The first one was to determine if acute and or chronic exposure to PFAS may disrupt ion movement and homeostasis, leading to disruptions in the electrophysiology of the cardiomyocyte. And the second one was to evaluate if the effects that we observed in some preliminary experiments in this I-cell line-derived cardiomyocytes can be similarly observed in, I like to call it like store-bought versus homemade cardiomyocytes. And so we make a house specialty. Our cardiomyocytes are pretty exceptional. So we're going to see if there is a similar between them, or if it's easier just to make it at home. And so my hypothesis is to see exposure to PFAS disrupts cardiomyocyte electrophysiology, and it can be observed with optical mapping. And so before we can really jump into cardiomyocytes, you have to ask yourself, where is the source of all of our cardiomyocytes? We're not just like harvesting them from people. That would be pretty cool, but it'd be kind of hard. So we get them from something called human-induced pluripotent stem cells. And what those are is they're cells that can be derived from an adult patient like you or I. They take your blood, they isolate these special somatic cells, and then they can use a virus that infects them with these special transcription factors known as the Yamanaka factors. And those include the ones listed on the screen. I'm not going to read them out. And these were discovered by this guy, Shinya Yamanaka, in 2000, and he won the Nobel Prize for 2012 because this was like huge. We got these stem cells. They're not embryonically derived, so there's less ethical questions and concerns, and we can do a lot of really cool stuff with them, which includes later on being found to also be able to differentiate these iPSCs, as I'll call them for the rest of the presentation, into cardiomyocytes. Oh, hold on. Sorry, I'm getting a phone call from Andre. Thanks. So here I'm going to walk you through a little bit of this complex protocol of actually differentiating and purifying the cardiomyocytes. So we start with that blue little hexagon there where we're maintaining our stem cells in colonies. And that blue background is that first plate that they're being cultured on. And so we'll pass that into, once they've reached like a high density, we'll pass that onto the second plate in the middle here, which is the orange and red, and we'll grow a tissue monolayer of stem cells. And then once there's a nice tissue monolayer of stem cells, we then begin the directed differentiation, which starts with cheer, which is pretty much it. We're going to start doing some epigenetic modification on these cells using these small molecules. So I'm sending signals saying, hey, it's time to differentiate into the mesoderm. And then cells say, okay, we'll differentiate into the mesoderm. It's that easy. So that's on day 0. And in day 2, we add another small molecule that inhibits a special pathway called the Wnt pathway. And so not go into the specifics of that because I don't know them, I'm going to be honest. And over the course of a few days, we maintain those on basal media after inhibiting the Wnt pathway. And then we're able to reactivate the Wnt pathway when we change from our basal media to an RPMI media supplemented with something called B27. And so after 8 days, and between 8 and 15 days, we start to observe these spontaneous contractions of the cardiomyocytes. And then we allow them to mature for some time. Between day 15 and day 21 is like an ideal time point for us to then take those cardiomyocytes and purify them so that it's just the cardiomyocytes. Because similar to a regular cell, or to the regular human heart, we have a mixture of different cell types that arise from differentiation. And that same thing happens here. However, we're just trying to isolate the cardiomyocyte. And so we use something called a magnetic bead separation. And we actually are able to use antibodies, magnetic beads, and magnetic fields to trap the cells that we don't want in the magnetic field and allow the cardiomyocytes to pass through. And that actually can yield a purity of up to 98% cardiomyocytes, which is super awesome. And it's not, and thankfully, because it's a negative selection, the cardiomyocytes themselves aren't getting put under, are getting put under less stress than the positive selection of the other cell types. So that means that we have a high efficacy of survival of those cells as well. And so once we've purified them and I've done that Herculean effort, we're at like 21 days. So it's been 3 weeks and I haven't messed up yet, and that's great. And so I wait another week post-purification to allow those cells to mature. So now we're about 30 days in, and then that's when we can start beginning our experiment. So this was a neat video of iPSC derived cardiomyocytes. I'm going to switch over and share a different screen, so you guys can see what these cardiomyocytes look like under a microscope. So you can kind of see here, there's like this little tight change happening, and I know it's very small, but under a microscope, we're able to see, we're able to kind of measure that that metric, or we're able to measure that. Oh, screen sharing is stopped. That's right. So those are what the cardiomyocytes look like, and it's pretty cool. I could literally sit there all day long, just watching those cells beat in a dish. It is like the greatest feeling in the world to be like, I made these human heart cells. And that feels just, like, I kind of have a little God complex maybe. Okay, so now it's time to poison the cells that I've taken so much care into taking care of, put so much effort into taking care of them. Now it's time to poison them, because that's why I raised them to begin with. I brought them into this world, I'm taking them out. So I use a few different controls. I use DMSO, I use doxorubicin, E4031. DMSO is a solvent. Doxorubicin is like the king of cardiac toxicants, and this thing will just destroy your heart cells. E4031 is a hERG channel blocker, so it blocks potassium. And that basically affects action potential duration, which affects cardiac electrophysiology. At one time, I accidentally gave my cells an ischemic heart failure, so they weren't beating at all because I dosed them with so much E4031. So now talking about the acute cardiac toxicants, we also have the carcinogens. So I use different concentrations of PFOA, as you can see listed here. And so I use between 0.5 and 500 micromolar with this increasing concentration. And so the cells have matured for a week post-purification. I start another day 0, which is the cells being dosed with PFOA. And I do that every other day until day 7. And that's when we go into data collection. So you can see my cells hanging out on the bottom of the plate. They're just beating, and then they're getting zapped with PFOA. So in the Heron lab and in the cardiovascular core, we use this special procedure called optical mapping. So we load this plate with the with our poison cells, and we're going to add this fluorescent dye. Most often we use Flovol, but there's which is a action potential sensitive dye, but we also have a special dye called Calbright, which is sensitive to calcium. And so what happens is Flovol emits a light relative to intracellular membrane potential, which is then collected by a high-speed camera. So from kind of like the overarching, from the actual, what it looks like on the benchtop, we shine a blue light that shines 488 nanometers, and that's going to be the excitation of the fluorescent dye. And when the fluorescent dye is excited, it's going to release green light. And so we fix our camera to just collect the green light that's emitted. And so we'll place the 96 well plate underneath the camera. We'll take a 10-second video. And what you get are this really cool video of different activations. So if you see these kinds of like little flashes in all the different wells, that is depolarization of this. That's depolarization happening in the monolayers. So that's action potential being propagated, and we can measure those changes in fluorescence. So that is really cool. That is like the bread and butter of the lab. So what's really great is after our 10-second video, we get this beat trace, which you can see here. And with that, we can do some data analysis. So we can get beat frequency, we can get the action potential duration at 80% repolarization. We can also get the triangulation of that action potential duration. We're also able to measure median conduction velocity, the time to peak, and upstroke slope. And those are just different phenotypic descriptions of the electrophysiology. So pretty much each of those peaks is kind of one change in action potential. And so a little bit more vocabulary for you. If you're not familiar, excuse me, we have regular beat frequency. That one's pretty obvious. APD is kind of a measure of repolarization. It's a measure of ion movements. It serves as a biomarker to compare if we see disruptions in ion movement and homeostasis. And we do that using APD 80 and the triangulation. Median conduction velocity is just the time it takes for the signal to travel from point A in the well to point B, because we have a whole well video, we can watch that change over time and we can measure the speed at which that happens. The time to peak is the time in which it takes from the start of contraction here at resting all the way to its peak here. And then the upstroke slope is just, so it's the time it takes, and then the upstroke slope is just the slope of how quickly that depolarization occurs. So I'll let that soak in for a second because it's a lot of information. Oh, and there's our guy again. There's those beat traces. So once I have generated all that data analysis, I gotta like slam it through Excel and Prism and all of my like fun statistical softwares. And so we take the average beat statistics of the acute or chronic, and we'll normalize those to the control. So all the data you're about to see, the effects will be relative to the control of that experiment. And then we do our statistical analysis. I run an ANOVA, do a post hoc, honestly significant differences test, and we get the data that we see today. So in 2020, one of our colleagues conducted preliminary experiments where they did a screen of different environmental chemicals that we believe to in fact affect cardiac electrophysiology. And so PFOA was one of those. And what did we see from that data? So we exposed the cells for 1 hour. We exposed I-cell cardiomyocytes, which were those store-bought ones, for 1 hour, and HBSS, which is just a salt solution. And it doesn't contain proteins that PFOA can bind to. That will be important for later. So just keep in mind that that there isn't proteins in HBSS, and it allows for pretty efficient delivery of PFAS to the cells. And what we saw is with increasing dose, we saw an increase in beat frequency. And at this highest dose, we saw a decrease in upstroke slope, a statistically significant decrease in upstroke slope. We didn't see effects in any of those other endpoints I mentioned, which were the action potential duration, triangulation, 80, time to peak, and median conduction velocity. And so in summary, from these first things, we saw 500 micromolars was really like, bam, that's really hitting them. So that was like hitting them with a cannonball, honestly, because that's a pretty high PFAS concentration. So what was really neat is to like look at those actual beat traces. And so you can see here on the right, you have the beat trace taken as like a baseline before this exact well was hit with 500 micromolar PFOA. And then 1 hour after treatment, you can really see that there is quite a particularly significant difference in behavior of the beating of the cells. And so that is like a pro-arrhythmic response. We also did a chronic exposure, and that was for 1 week. And these cells, this PFAS was dissolved in a media that contained proteins. So I think it's kind of more similar to what you would maybe see in a human, because the main compartment of PFAS is in our blood. And so it only makes sense that using a media that also has a lot of proteins similar to humans, like the PFAS is bound up in our proteins in our blood, to do our exposures using this same media. And so what we saw in these chronic week-long experiments is that we saw effects in action potential duration. We saw it in the triangulation and the repolarization at 80%, where we didn't see effects in these other endpoints. And so what's interesting is that you have a shortening of action potential duration, and I'll discuss what this means later. But that's like a pretty significant, like very significant to like have your action potential duration cut in half. And so you can see that here that like each of those peaks, like there may be the same level of beats and it may be a similar change in fluorescence, but the duration for which those beats is occurring, you can see like it's kind of like the triangle, the base of the triangle is kind of been squished. And so that's how we can see that there are changes in action potential duration with these fairly resilient cells, because whatever cells are left, they're pretty strong at this concentration. So from these preliminary results, we see that there are effects in the acute and chronic exposure to PFOA. And there are various endpoints that are pointing to upstroke slope, beat frequency, and action potential duration. And so that was kind of great that I was able to even get that data to begin with. That took a little while to even just like extract, learn all the software, and kind of with that, with this data sitting, sitting in front of me, I was like, all right, I think I can handle this. I want a shot at this. I want to see if I can do this same, if I can like beef up this experiment, try again, or try it all. And wow, that was quite the experience. That's, yeah. Let's go into that experience. So I conducted chronic exposures. The first one was on, again, those store-bought cardiomyocytes. So I wasn't differentiating stem cells into cardiomyocytes yet. I was still practicing that, but I was really excited to at least try to get an exposure done. So again, we did that same chronic paradigm, but I added a few doses, right? Because toxicology, I'm trying to get more of a dose response. So I squeezed a few more doses in there. And what was great to find is that we see, again, effects on action potential duration, 80, and triangulation, similar to the exposure that happened 2 years ago. The caveat is, then there's no effects on these other endpoints here. Okay, well, yeah, that's a significant effect there. The caveat is that these are different directions, which is a really interesting thing. So instead of seeing a shortening, we see a prolongation of action potential duration. And so that was a very interesting discovery, and we'll talk about that more later too. Shouldn't be leading with all these caveats. So this is another exposure. That was the first chronic exposure. This is a second chronic exposure. Luckily, I'm sorry. This is the same data from this experiment. But I didn't see a significant effect on upstroke slope when we ran in ANOVA. But what was interesting though is that when you take when you transform the PFOA concentration into its log base 10, and then do a linear regression of 0.5 to 50, you actually do see a statistically significant increase. It's a very weak R squared, however, it is significant, and that was great. And so I think in the future, it would be great to power this study a little bit more. And that was also, again, with our store-bought cardiomyocytes. Now, after some time, that was some time, we looked at the data, kind of pondered over things. And in that time of development, I finally, months and months and months, was able to differentiate these cardiomyocytes, purify them, get them so that they were beating, and get them set up, ready to go for my own experiment, all homemade, so that's great. And so when I ran this experiment, I wanted to know just that I saw one of the first things I noticed when I was dosing these cells after a week were these changes in media color. And if you've worked in cell culture before, you know that you obviously want to be watching out for media. So what I found was a really interesting kind of monochromatic scale where as soon as you kind of cross this threshold from 50 to 500, you see, on the left side, you see this light pink. That's really normal for a few days. If you're changing media every 2 days, it's normal for it to change that color. But from 50 to 500, that looks more like media was just changed in it. And like that kind of indicates that that there's probably less like metabolic activity, and that lowering of metabolic activity is connected to the amount of cells that are actually surviving. So I've read in literature and in my own personal experience, it seems that like 50 micromolar is when this concentration gets so high that you're not even affecting like electrophysiology. You're just killing those cells. You're just like, you're affecting electrophysiology, but you're also just killing them. And it's great that we're killing them, but then you're measuring a different effect. So I've kind of, this experience here, just looking at this kind of encouraged me to narrow down in between the 0.5 and 50 micromolar. And so with those homemade cardiomyocytes, I'm sorry, I'm calling them homemade, I conducted a chronic exposure, and what did we see again? We didn't see any significant effects except one, which was in the upstroke slope. We do see a significant effect with the ANOVA. I conducted a linear regression again. This time the p-value was more significant and the R-squared was slightly higher. And so that was great to kind of see this corroboration of like, okay, there's an effect happening and it's kind of like starting to like be paired. And if you're still a little skeptical, I did it again. And I did it this time. So that was in one cell line. I did it again, a few different experimental parameters. This time I did an acute exposure with our contemporary PFAS compound of interest. And instead of using a fluorescent dye, action potential sodium dependent fluorescent dye, I used a calcium one instead. So the beat traces are going to be slightly different, but still kind of indicate to us that there's some kind of disruption in ion homeostasis happening. And what's great and not great is that GenX seems to be more effective at lower concentrations than PFOA. And again, we see this dose-dependent change from 0.5 to 50. This is actually from 0.5 to 25, where we see significant increase in the upstroke slope. R-squared is much more powerful and it has a higher p-value. So it's kind of great to kind of see this corroboration of like, I'm seeing upstroke slope, So that will make me want to change, that will make me want to continue to focus on this is a potential endpoint in future experiments. So I just blasted so much information. And when I was going through it, I was like, I don't even know how to keep track. So I made this table for myself and was like, you know what, these people might like it too. So from the experiments, both past and present, this is a summary of where we see significant effects happening. We see some in action potential duration. We see some in beat frequency. The upstroke slope is really hitting. And we didn't really see any changes in time to peak and median conduction velocity. And I find it interesting that we didn't see significant effects on time to peak, but did on upstroke slope, although they do have a relationship with one another. So kind of thinking about, all right, well, we see effects in action potential duration, we see effects in upstroke slope. What does that mean? What does that mean for anybody? What does that mean for the public, for public health, environmental health in general? So action potential prolongation can induce something called Torsades-Dupont, and that's a form of tachycardia that can lead to cardiac death. And this is associated with potassium ion channel signaling, the flow of potassium into the cells, and intracellular concentrations of calcium and sodium. The upstroke slope is driven by ion function, by ion channeling. And after this buildup of action potential, you have the potassium that's going in to be like, all right, calm down, we don't need this much. And so these changes in action potential can lead to abnormal cell function, where we saw like these funky beat traces, and those could be precursors to an arrhythmia. And so I think a potential target for that is effects on the gap junction, or the rate of flow of calcium in and out of the cell, as PFOA may be like affecting the membranes, making them more leaky. We're not really sure, but obviously we want to continue to dive in on how these ion transporters are being affected, gap junction function, and transients of like calcium and sodium ions inside of the cells. So this is, again, just an inventory of things of past, present, and future. So I've done we have some technical replicates. These needed replicates are kind of what I think we need to continue to push the science further, to kind of just corroborate and say, all right, we saw effects once, can we do it again? And it's like, all right, I'm still skeptical. Maybe we should do it one more time. We'll see. And so this is kind of where we're going for the future. I also just wanted to kind of characterize and thinking about concentrations in the general public versus those exposed workers versus the doses in my experiments, because that's something that's kind of, I think, needs to be more important and transparent in toxicology. So the blue bars are from an NHANES data set, and that's biomonitoring studies from 1999 to 2017. I also had a biomonitoring study of retired 3M workers, which were those guys who are those people who were generating using PFOA in industrial processes and in manufacturing. And those are the yellow bars. And then green is was chosen by our team as doses to hit them with. So you see that there's an overlap in between workers and the doses we're using these experiments. But for the general population, I also want to note that the y-axis is log scale. So these are orders of magnitude difference in concentrations. So in conclusion, PFOA exposure has been found to cause disruptions of electrophysiology in iPSC-derived cardiomyocytes in both the acute and chronic treatments. Significant effects were observed at this most high dose. But what that kind of means, though, is that it takes 500 micromolar to really see an effect. But so we know that there are a section of cells that can be very adult cardiomyocytes that can be resilient to high PFAS exposure, which is really neat. Some of these toxic outcomes that I mentioned earlier, where we saw a prolongation in one experiment and a reduction in another, could be associated with the type of matrix that we use. So we use two different types. We use Matrix Plus, which is an extracellular matrix, and PDMS, which is this soft silicone matrix. I actually just learned today that PDMS is also used in environmental sampling and detection of PFAS in the environment. So it has a high propensity to sort PFAS to it. So that also is a modulator of our experiments, as well as the media type and PFAS's availability in a media that doesn't have proteins. So PFAS is just floating around freely versus a media that has protein where the PFAS is bound to the protein. And so based on what I initially found, it seems like PFOA may not be deleterious to the population as a whole in terms of its electrophysiology. However, workers may be a different story, but we'll continue to kind of probe that. So some next steps, again, is to replicate some experiments using the different voltage sensitive dyes, to do different endpoints other than just electrophysiology, and to look at PCR, to do PCR and Western blots, to examine genes being expressed, messages being sent, and the eventual proteins being expressed. And then once we have all of that huge stack of data, it would be great to just bring it all together and compare those results and see, if we use stem cell lines that are derived from different sexes, do we see different responses in those sexes? And do we see that in the physiological endpoints, biochemical or genetic responses? So some future directions, it'd be so cool to run the differentiation while exposing the cells to PFOA, because we know that this is a developmental toxicant and it can affect embryogenesis. It's been associated with, again, that decreased birth weight and different developmental organs or disruption of certain organ development. So it'd be great to see if we can run our model and see how the cells are behaving not exposed to PFAS versus being exposed to PFAS during differentiation. So now we think, all right, this guy's getting a master's degree in environmental health, so I should probably mention a little bit more about environmental health. It's impossible to avoid PFAS exposure. I'm sorry. And we're exposed to 10 to 12,000 different ones, or there's 10 to 12,000 ones out there. So you can probably safely assume you've been exposed to a decent amount of those. I don't know why we still use highly fluorinated carbons in manufacturing at all. I mean, I do know why, but from an environmental health perspective, it is not a good idea that we still use PFAS, and it needs to be phased out ASAP. And at the general population level, again, it may not be sufficient to precipitate cardiovascular disease on its own, but overall, we're depleting the body's physiological reserves. We're increasing the burden with all these other exposures that we get through across our lifetime, and that may kind of push us into a disease state and keep us there longer. So With that, I want to just acknowledge some people. I want to acknowledge Tvoko, who's been like my pal, my buddy, my teammate on keeping these cells alive, differentiating them. We've been partners in crime. I want to thank my committee chair, Dr. Sabota. I want to thank Dr. Justin Colosino. I want to thank Dr. Andre Montaldo de Rocha. And I want to thank Todd Heron, who is on the Zoom. I also want to thank the Do Goods Pie Lab. I want to thank Emily for giving us some money. I want to thank Westwood residents advocating for themselves, because without this organization, I wouldn't be here talking all crazy about PFAS. So shout out to them. And then I want to thank my alma mater, UMass, because the people out there were just super supportive. And without them, I also would not be here being able to talk like a scientist. And yeah, overall, thanks School of Public Health. Thank you, my family. I love all of you all so much, my friends. And that's all I've got for you today. Questioning from the general audience. Yeah.
So about PFAS binding to proteins.
Mhm.
So I have a couple questions in that realm. First of all, are the NIEHS measures, or not the NIEHS measures, the national measures of PFAS, are they measuring bound or unbound PFAS in blood?
That's a great question.
All right.
I know they're measuring it in PFAS, the serum of blood. So I imagine if it's the serum, it's probably what's bound up in the proteins.
So also then follow up, do you know what the unbound concentration of PFAS or PFOS, I guess in this case, was in your particular cell cultures?
Nope.
Okay.
Or I do know the bound, I don't know the unbound.
Oh, well, then-
Or I'm sorry, I don't know the bound. I do know the unbound, because that's just like relative, theoretically, it would just be whatever concentration we dilute to, but what's available to the cells. There's we've had a lot of debate on like the fate and transport inside of like this, you know, if you have this little model of like, where's the PFAS going? Is it sorbing to the plastic? Is it sorbing to the cells? If it's bound up in the proteins, are the proteins like the source and like the truck driver that's like delivering the cell, like the PFAS directly to the cell? Is the uptake more available when it's free when it's in free, you know, free floating space or bound to the proteins? They're more available in free floating space. Those cells are getting hit way harder than the ones that are just maintained in media that contains proteins.
so how do you think the actual like levels compare for like 50 versus 500 versus in the samples that you have media with binding proteins going on?
You know, I wish I was a chemical person, or like I am a chemical person, but like a physical chemist, because they would probably have a great time with answering your questions. And I'm honestly not sure. Yeah, thank you.
Yeah, I just want to know, this is something that we've been grappling with, and I've been talking to Sue Fenton and her team at the NIH. And of course, for those of you who don't know her, she's a huge PFAS person. And they actually have we've started working with the same protocol she works with, which is to actually pulse the PFAS in PBS or HBSS without the proteins first for a half an hour. Is it half an hour or an hour? We do it.
We do an hour.
We do an hour, and then add in the medium with the proteins, so that not all of the PFAS is just getting gobbled up by those proteins. Because yes, in the body we have PFAS that's bound to proteins, but we also have it in our body for years, and so there's more time for the PFAS in our body to get to our cells. Whereas in a cell culture experiment, if you're doing an experiment for an hour or even 7 days, it's harder to replicate.
And that this is even related to a question that Monica Roy posted in the chat. Would your exposure setup be considered an acute or short-term exposure? I wonder that even though you didn't see the significant effects of your lower concentrations, if there might be effects over the long term. I don't know much about the cardiomyocyte experiments. Was there any way to study the effects of chronic exposures with this model? So I've been just like hanging out and just like keeping some cardiomyocytes alive, like, and they've just been sitting there, and like they can hang out for like a couple months in culture, probably even longer depending on the user, really.
Yeah, you can keep them in culture for years.
Yeah, as long as you don't screw up. So yeah, Monica, so to answer your question, I guess theoretically we could I've even thought of what if we, you expose the cells Monday through Friday, and then just like a worker, you take them off the exposure Saturday, Sunday, and then start back up again. And what if we just kept doing that over and over again? Even at these low doses, is it enough to really tip them into a more disease state? But that's a great question and consideration. How long is a long-term exposure? Because some people will do cardiomyocyte exposures for 5 to 6 weeks versus 1 week. This is such a developing model that we still don't even have a shared agreement on what acute versus chronic exposure means. And Todd is already jumping in. Do you think that the effects will be the same in fetal hearts and adult hearts? Can this be modeled in vitro using iPSC derived cardiomyocytes? So that's a great question, Todd. So yes, in some ways it can be modeled that way. When we plate our differentiated cardiomyocytes, we have these two different matrices. We have, like I mentioned, matrix plus, and we have PDMS. And when we allow both those cells, when we allow the same cardiomyocyte to mature on PDMS versus matrix plus, we see that they behave a little differently in their electrophysiology, and even further than that in their actual biochemical and protein expression as well. And it all has to do with just the softness of the plate and of the actual matrix that they're sitting on. And so what we find is that the electrophysiology of cells plated on PDMS, although they're mature, they have a more fetal-like phenotype, versus matrix plus has this much more adult-like phenotype. And in terms of assessing, for example, chemotherapy agents and the effects that it may have on the heart, we want our cardiomyocytes to behave like adults, because adults are the ones getting those chemotherapy agents. So we want to definitely consider how these differences are observed. And that could also be why you see this uptick in action potential duration one way, and then it's downtick in action potential duration in the other, because of the different matrices that those are plated on. So thanks for your question, Tom. Yeah.
Yeah. So great talk. It was very interesting. So with PFAS, you used calcium to measure the action potential change, correct?
It's a membrane potential sensitive dye. So it's not
okay.
just calcium that's being observed. One of the experiments, the last experiment I showed, was calcium sensitive, but the rest of them are sensitive to pretty much the membrane potential, which is all ions.
Oh, okay. For some reason, I thought it was more sodium sensitive. And I was just wondering, specifically by using that measure, did you see a significant difference in your previous experiment when using the general measurement versus the calcium measurement, or why did you decide to switch?
So actually the last experiment that I showed you was actually the first experiment I ever did. And I had no idea what I was doing. So I was I'll just use this dye, and we'll use these concentrations. So I was just it was kind of very early on, just trying things out. And so I guess I missed your initial question. I'm so sorry.
Oh, it's more when you decided to switch what you were looking at. Did you see a difference, or are they very similar?
The signals that you get from the two dyes, the measurements are going to be different, because it's you're measuring different metrics. It's telling you a similar story, but it's you're reading the story in Spanish versus English, I guess, maybe I'd say. I don't know if that's a good analogy or not. But
yeah,
it's
there's a little bit of translation that's missed, but it's still the same thing.
Yeah, right. You're going to get the story. No,
it's not the same thing.
No? Okay.
No.
Then.
So the action potentials, they precede the contraction, and the calcium changes that you're seeing are triggered by the change in voltage. So there is something in electrophysiology that's called EC coupling, the electrophysiology contraction coupling. So when you're doing voltage, you're checking the electrophysiology parts that triggers the contraction. So they're different things.
So maybe
they are interrelated. Let's say that they're different chapters of the same book.
We'll work on this. We'll work on that analogy. We'll work on that analogy. Thank you, Andre.
Right, and then I think my other question was, so you're growing these cells and then you sort them, so specifically you have one cell type, correct?
Mostly, yeah.
Did you have a control where you didn't select for that? What was that? Did you have that control in there where you didn't sort?
That's a great question. That's a great question and recommendation. We don't. Usually what ends up happening is we have all of our cell types, like you have like the cardiomyocytes and then everything else. And the focus has been so heavily fixated on the cardiomyocyte that we kind of ignore the other cell types. So it'd be great, I mean, honestly, because they also are part of the heart and, a function of measuring toxicity. So that's a great question. We don't include those in our control. We don't have a control for that, but that would be a pretty neat thing to try to maybe consider in the future for experiments. I have another question from Todd, and then I'll take another one from the audience. Do you think the effects will be the same in fetal hearts and adult hearts? Can this, wait, did I just, it's the same question. Sorry, I thought it was another question. I just changed the size. So yeah, Rebecca.
Someone else.
Or-
Anyone else? Okay. Did you have any markers? I know that you talked about liability potential issues with your higher doses. Did you actually measure?
Not yet. So I've looked, I've looked at the cells and I know that they're like not doing great when you expose them to 500 micromolar. I've looked at potentially conducting some ELISAs to look at biomarkers of like lactase, dehydrogenase being excreted from the cells. So with LDH, you would at least be able to kind of measure a function of like, are these cells like becoming leaky because the PFAS is kind of, for some reason, stressing them out or changing the membrane. And it's as simple as just taking some media and then like allowing that media to interact with another fluorescent dye. And so we could use that to measure, you could use that as an association of like cell viability. I'd also love to do like an apoptosis necrosis assay as well. Just a simple, just like imaging. So those are two assays, like biomarker assays that I'd love to try. Yeah.
You mentioned that it would be interesting to do sort of sex-specific outcomes of this. I don't know anything about stem cells. Are they all like male or are they a mix?
So you can derive them from male or females, and then from that from that patient derived, like that patient specific derived line, if we had 3 stem cell lines from like that contain XX chromosomes versus XY chromosomes, we differentiate those cells and then we characterize the response. Hopefully from there we'd be able to start to see if there's any differences in response based on sex.
What about the work that you've done so far?
So I'm not sure what sex the I-cell cardiomyocytes are, but we have 2 cell lines that we've currently been using. The 258, which was the data from this experiment. This 258 was an experiment. And then like way early on, like one of the first times I tried a differentiation, it like worked amazingly and everything worked out. And then 4 months later, like I wasn't able to get anything to work again. So this 442 line is a female line, the 258 line is a male line. So to kind of like do the experiments that have happened in both of those, like with the opposite, and then trying to see and compare. I know it's only an N of 2. I don't know how many you really need to see sex specific effects, but it's at least a starting place to like start with physiology and then like climb our way down deeper into the levels of organization. Other questions?
So you mentioned about the individual. So do you think that the individual difference between different individuals might have a difference between?
Absolutely, like interperson, like uncertainty of like different genetic responses. That's probably, that's one of the harder parts of using the cells is that, like to really get certain, this biological replicates, although each well is considered its own replicate, compared to like an animal model where you get a diversity in genetic, like you get more genetic variance, like theoretically in this cardiomyocyte, it's still from one person. So you don't get that same level of genetic variance. So that's again why we would need to test it in multiple cell lines. Again, the benefit of using the iPSC, the human iPSC line is that it's human, so you don't have the interspecies comparison, although it's still cells grown on a plastic dish, so there's like that difference between like, my heart and the hearts that we grow in our plates. But yeah, of course, that uncertainty factor is still there for some part.
Chris, just think that most of the studies with animals, they use isogenic lines, isogenic strains of mice.
So they're just very.
So they're basically the same thing.
Okay.
Do you think it'd be worth exploring a PFAS mixture effect on electrophysiology to see if at low doses together do something?
Absolutely. And so I ran an experiment early on as well when I was like, I don't know what I'm doing. I'm just going to do stuff. I actually dosed the cardiomyocytes with a low concentration of PFOA, one that I didn't think would elicit an effect, and then dosed them with GenX as well. And I thought I would get like some kind of effect. But I didn't really see anything like very clear and distinctive, but it would be wonderful if we can get this, model, like if I can adapt to the model, because it's more of like us like adapting to being able to use the model to get it to a high throughput like state where you could then assess multiple chemicals and multiple mixtures at once would be super awesome. Absolutely be a great place to go. And you can definitely do it in this model. It's just dependent on the amount of time you have.
Yeah.
And the results of that experiment too were confounded by this binding of the PFAS to the serum and to the proteins in the cell medium. So we don't even know if the cells were seeing any of it at all. And yeah, so some of these effects that we're seeing or not seeing, maybe an underestimation of the true effect. It's difficult to fully say because again, trying to take what we have in cell culture and relate that back to what's happening in humans with these differences in the exposure time and the protein binding and everything and the mixtures that we have, in our bloodstream compared to, just using a couple in a dish, it's complicated.
Yeah, I was wondering, I know you had mentioned studying developmental effects as the cardiomyocytes were matured. Would you also consider studying it as the stem cells themselves were derived into cardiomyocytes, like during embryonic stages and then as the cells were forming?
Definitely. Yeah, that's, I should have been more clear. That's what I think I really mean is like, when we start like our culturing and when we like start plating that monolayer, like before we even start the directed differentiation, expose the stem cells, expose them during directed differentiation, and then seeing how they just behave when they come out the other side would be a fascinating exposure paradigm and experiment to look at.
Another question. So is there a way to induce stress to the cells so that you could see a phenotype?
Yep.
Did you do that? And
I did
how
not.
do you do it?
That is
Okay.
another thing we're thinking of doing.
Yes.
So we have a wide variety of drugs that interact with cardiomyocytes. E4031 and doxorubicin are two like ones that we could use. Isoproterenol is another one. So there's a wide variety of like pharmaceutical approaches that we can take like in combination with like our toxicological ones. So it's like, if we dose these cells like and then hit them with a stressor, how is their resilience different? Based on like not like with and without the stressor. And is PFAS plus a stressor then enough to precipitate some kind of adverse outcome? Yeah, absolutely. That is definitely an area we
That'd be
want to
really
study
neat.
too. There's so many options. There's like so much we could do. And it's
Pick
like,
one.
how much time do you, unfortunately, we're not on this earth forever. So can't ask all the questions.
Yeah, that's something that we're really interested in my research. One of my research goals in the iPS cell model and in the mouse model is, just at baseline, maybe there's no toxicity, but at some secondary hit, like a stressor, your poor diet, you have a terrible, some sort of life stress, you have pharmaceutical agents, other exposures. Does that bring out the effect?
Yeah.
Any other questions?
One last one.
Yeah.
Did you do any assessment of potential outliers on any of your examinations? And did you correct for multiple testing? Yeah. Okay.
Would love to have your statistical guidance. I reached out to someone from the MLead Center who was like a biostatistician, and they're like, "Are you able to meet on Friday?" And I'm like,
Oh, dang.
I'm graduating on Friday, so couldn't do that. But there is so many statistical tests that I want to do, that I just don't have that experience. And so it'd be great to like continue to build up our N, so we have more statistical power to begin with, and like reproduce, like be able to say like, we ran these 2 experiments, everything was theoretically the same, except, you know, the time in which we ran them, and then being able to like do more hard statistics on that would be awesome.
Great job, Chris.
Thanks so much. Anybody else?
So Christopher, what like excites you most about like, doing all the research and the experiments, and like how do you see all the research you've been doing? Like what are your long-term goals with this?
Okay, yeah. Well, it excites me because it has to do with PFAS, and I really don't care about anything else other than studying PFAS. And so learning this whole model was like an ends to a means to study PFAS. No, it wasn't really fun. It was really fun to learn and understand this model. And I think that I found that what really excites me is that like I can get like this black box, like the model itself, and like have my PFAS and like put it into the black box, and then it spits out all of these results, and like trying to understand how that black box works, whether it's in cells or in zebrafish, or whether, you know, one day it's going to be like in little microbes or whatever the model is. I think it's really fascinating and fun to like try to come to a higher level understanding of biology through just like technical approaches, because I don't have a formal biology education. Like I took a few bio classes, but like all the biology I've learned is through my mentors. And I think learning is just so addictive. So that's what really motivates me. So I'm hoping to just like be a professional student and just keep learning and teaching people about PFAS and how screwed up it is. So great question. Thank you all so much for coming.
Thank you all so much for attending. This concludes the public portion of the defense. So we'll just have a session with the committee members and Chris.
Mhm.
And
Yeah, if people want to, I bought a bunch of snacks. I would love for folks to hang out for a little bit, maybe in the cafe. My mom's got the bag of snacks. So if folks want to like hang out in the cafe, if you're not going anywhere, I'd love to like chat with people when we're done with the more intense stuff. So thanks again, everybody. Thanks, Chief. Yeah.
okay.
For sure. See you tomorrow.
Yeah.